In this video, we'll be talking about chemical synapses and looking at neurotransmitters. So, typically, when we're looking at a neuron, we have a presynaptic neuron, which then interacts with a postsynaptic neuron. And of course, the space in between is a synapse. And there's real advantages to using a synapse. It gives you more control in terms of the message versus something like a gap junction, etc. And typically, what we're looking at is a release of neurotransmitters that will interact with an appropriate receptor on the postsynaptic neuron. And typically, what we see is that the axon terminal of a presynaptic neuron will contain vesicles containing a neurotransmitter. And when stimulated, the presynaptic neuron, so if a action potential travels down the membrane of presynaptic neuron, it then causes the vesicles with the stored neurotransmitters to move to the surface and release them. So we have many vesicles that are docked on release sites in the plasma membrane, ready to release a neurotransmitter. And as I said, the postsynaptic neuron will have appropriate receptors for that neurotransmitter. And because we're talking about neurotransmitter binding on to a gate, we're looking at ligand gates. Now, let's remind ourselves that, as we had in some class discussions when we were chatting, is that it's very typical that a postsynaptic neuron will actually be interacting with a whole number of presynaptic neurons, and that gets back into the concepts of the summation in terms of spatial and temporal summations. But let's spend some time talking about the structure of a chemical synapse. And as I said, down here in the synaptic knob, we have a number of synapses that already have pre-made neurotransmitters. And the typical way this works is an action potential will come down the membrane of the presynaptic neuron. And often what the trigger is, it opens a gate. And in many cases, this is a calcium gate. And calcium floods in. And calcium allows the vesicles to basically bind with the cell membrane in the presynaptic neuron, and with that fusion of the vesicle, we basically get the discharge or release of the neurotransmitter, which will go across and interact with the ligand of the postsynaptic neuron. Now let's have a look at the classes and the categories. There are quite a few known neurotransmitters, and we don't certainly know them all, but there's more than 100 neurotransmitters have been identified to date. But we can break them into a few major groups. And the four groups that we are looking at are the acetylcholine-based ones, and it's actually a small group. We're going to see it only has one member in that group. The ones that are based are amino acids. They are amino acids. We have the monoamine group, and we have the neuropeptide ones. So let's start off talking about the first group, which is the acetylcholine one. Now, this is an easy one because the only member in this group is acetylcholine itself, so it's named after the neurotransmitter that's in there. And it's formed from acetic acid and choline, thus its name. Now, the other groups we'll just introduce briefly. We have the amino acid neurotransmitters, and if you're familiar with the names of any of the amino acids, you can see they're represented here. Is that we have things like glycine, glutamate, aspartate, and this is gamma-aminobutyric acid. Now, that is often just referred to as GABA, All right, next group would be the monoamines. And the monoamines are made from amino acids. And some of the major ones within this group are epinephrine, norepinephrine, and dopamine. Now, just a little side note that epinephrine is also known as adrenaline, and norepinephrine is also known as noradrenaline. Histamine, ATP, and serotonin can actually fall within these groups. Another group we have are the neuropeptides, and the name tells you a lot. It's basically peptides from 2 to 40 amino acids long. And they're stored in secretory granules, and some examples of those would be things like cholecystokinin, which we'll talk about in future chapters, and substance P. Here's just a quick look at some of the groups once again. There's the acetylcholine. There's only one member in the group. It's ACH itself. We have the amino acid group of neurotransmitters, and of course GABA is one we will highlight. Glycine, aspartic acid, and glutamic acid are some other examples. In the monoamines, we have epinephrine, norepinephrine, and dopamine, serotonin, and histamine. And here we can see some examples of the neuropeptides. And you can see they're just strings of the amino acids together. And some examples would be things like enkephalin, substance P, cholecystokinin, and beta endorphin are some examples of those groups. There's also the gases, and it's kind of odd to think that sometimes things like nitric oxide and carbon monoxide have a role in here. And these are inorganic exceptions to what we usually think of as neurotransmitters. Now, these are interesting because they're synthesized as needed, and they don't get stored in synaptic vesicles, and they diffuse out of the axon terminal. So it's not a release by exocytosis, and they diffuse to the post-synaptic neuron. And they don't need to bind onto a membrane surface receptor. Also, we have the purines, adenosine and ATP also have a role. Now, synapses are variable. Often we'll consider them as excitatory or possibly inhibitory. That sometimes the signal sent from a presynaptic neuron to a postsynaptic neuron is an attempt to get the postsynaptic neuron to depolarize and form an action potential. In other cases, a presynaptic neuron may be trying to inhibit a postsynaptic neuron from actually firing. So some neurons are excitatory, some are inhibitory. And sometimes the transmitter's effect differs depending on the type of receptor. And this is something that we're going to see in this chapter and also see it in the endocrine system, is that often it's not the chemical messenger who determines what's going to happen at its target. It's the receptor at the target, that you can have the same chemical being sent to cells, yet on one cell the receptor is in such a form that it'll interpret it in one way, yet on a different cell, a different receptor, which will accept that chemical message or the neurotransmitter, will act in a different way. So it's not always the messenger, it's also the receptors who can have an influence as to what the effect will be within that cell. But some receptors are ligand-gated channels, and some act through second messengers, systems that we've seen before. Now we can have a look at some of the major kinds of synapses that we can come across. So let's have a look at examples of three kinds of synapses. We have the excitatory cholinergic. Now when you hear cholinergic, think about the name. It sounds like acetylcholine, and you're right, it is. We have the inhibitory GABAergic synapses. And we also have the excitatory adrenergic receptors. Or synapses, I should say. So we have three types. We have some excitatory cholinergic, inhibitory GABAergic, and excitatory adrenergic synapses. Now, the cholinergic synapses, just think of the name, not surprisingly, they use acetylcholine. And typically what happens is an action potential comes down the axon terminal. It opens a voltage-gated channel for calcium. Calcium enters the cell and causes the release of the ACH. And ACH will diffuse across the synapse. So ACH diffuses across the cleft and binds to the postsynaptic receptor. And the ACH receptors are ligand-gated ion channels that either allow for sodium or potassium to cross the membrane. And of course, the entry of, say, sodium into a postsynaptic neuron will start to depolarize it. And as we've seen in the past, if you can get enough depolarization in the trigger zone of a postsynaptic neuron, I'm going to form an action potential. And we can see the process right here. That here, if an action potential sweeps down the membrane of the presynaptic neuron, this is a voltage regulated calcium gate. So the action potential will cause the opening of the calcium gate. Calcium will rush in, and the role of calcium is to basically, through a number of steps, is to get the vesicle to basically fuse with the cell membrane, and then I get a release of the ACH. Now, as the ACH is released, it's going to bind onto an appropriate ligand receptor. Let's say in this case, it's a sodium channel, and sodium will rush in, leading to potentially a depolarization. Now, since we're here, we should mention that it would be a really bad idea if the neurotransmitter ACH stayed permanently bound onto here, because the gate would remain open. So the neurotransmitter only binds very quickly and then will leave. Now the other issue is you have to be careful about leaving ACH in the synapse after the desired effect is achieved. So somehow I have to get rid of neurotransmitters that have been released. And in the case of ACH, one of the mechanisms is there is an enzyme, acetylcholine esterase, that's what this stands for, which takes ACH and breaks it down into two parts, the choline and the acetate. Now the choline gets picked up and recycled back into the presynaptic neuron for basically being recycled and rebuilt into a usable form. Now, let's have a look at GABAergic. They use gamma-aminobutyric acid, which is shorthanded as GABA. Almost everyone calls it GABA, but you should know the full name. And what happens here is the action potential triggers a release of GABA into the synaptic cleft. And GABA receptors are chloride channels. So not surprisingly, if I open a chloride channel, chloride rushes into the postsynaptic neuron, and it's going to start to hyperpolarize the postsynaptic neuron. So I get a hyperpolarization, which means that neuron is less likely to fire. So you can view it that the postsynaptic neuron is inhibited, much less likely to fire. Now, if I look at adrenergic synapses, they use norepinephrine. Now, just a reminder is that the old name or the other name for epinephrine is adrenaline. So you notice up here, adrenergic, that refers to the old name of adrenaline. And that helps remind me what an adrenergic synapse is, because the old name for epinephrine is adrenaline, so adrenergic go hand in hand. So I'm talking about norepinephrine. And as I said, norepinephrine is also called noradrenaline. And in this case, what happens is that the monoamines and the neuropeptides, what they can do is they can actually bind to a G protein that's coupled to receptors on the postsynaptic membrane. And we end up with an activation of the second messenger system. Now, hopefully you remember the second messenger system. So when I activate the second messenger system, I produce cyclic AMP. Now, as you remember, there's a number of steps in the second messenger system. So you may be wondering, why would I use a system like this? Because it seems like a very slow way to do things rather than just opening a gate directly and having the effect occur. Well, there's advantages, disadvantages. Is that it is slower to respond than cholinergic and GABAergic synapses. But there is an advantage, is that it gives you something called enzyme amplification. And enzyme amplification. And what that means is that a single molecule of norepinephrine can actually result in a vast numbers of product molecules. Because remember, in the second messenger system is eventually you turn on basically kinases, and kinases can do the job over and over and over, almost like a photocopier printing out the product that you want. So one neurotransmitter, or very few molecules of neurotransmitter, can result in a lot of copies of whatever you want to have happen inside that cell. And we can see an example here, and this is just the basic second messenger system. When the signal comes down, the neurotransmitters are released. In this case, they're binding onto a receptor. I get activation of the G protein. I get adenylate cyclase interacting with it. I end up with cyclic AMP, and the cyclic AMP, depending on the cell and a few other things, can do enzyme amplification, and that can lead to metabolic changes in a cell. Or the other thing it could do is it may be part of a pathway that results in a ligand being made, and ligand-gated channels are opened. Now here's the difference, is in this case, the ligand is basically inside the cell and gets activated or made and will bind inside the cell and open the gate from the inside, and then you'll have things like sodium rush in. So it takes a little longer, but you can have a much bigger, much more dramatic effect when you use this system. Now, how do you turn off the system? Because you can't let it go on forever. The neurotransmitter stays bound to the receptor for about 1 millisecond, and then it detaches. Now, if the presynaptic cell keeps sending signals and keeps releasing neurotransmitter, then the postsynaptic neuron will be continuously stimulated. But one way I can stop this is I just shut down the presynaptic cell. So the presynaptic cell stops sending or releasing the neurotransmitter. And then the neurotransmitter that happens to already be in the synapse, I can remove it or deactivate it. So what are the ways I can deal with it that way? So how do I end the signal? So I can do degradation. And in that case, I come along and I can break down the neurotransmitter that's in the synapse. So an example would be acetylcholinesterase, which breaks down acetylcholine into both choline and acetate. So an enzyme will actually cleave the neurotransmitter in that synapse, so it's no longer usable. The other thing that can happen is I could have reuptake. So the neurotransmitter, or if it's being broken down into products, gets absorbed into the axon terminal. And we actually see that we actually see both these processes can occur, is that I can break down ACH into its parts, and then part of the ACH that I have basically gets recycled. So I can do degradation, I can do reuptake, and the other thing that happens is I could have diffusion. The neurotransmitter or the broken down products simply diffuse away, so they're no longer available to stimulate the postsynaptic neuron. So it's just a matter of the neurotransmitter simply diffuses away into the local extracellular fluid. Okay, we're going to leave it there, and we'll pick up things like neuromodulators in the next video.